Hello, my name is Armand, and for my practicum experience, I worked with the UMD Fire Cell X team over the summer. The practicum experience was done on UMD campus inside of the microbiology building. The overall research goal for my practicum was to clone human ribosomal proteins into bacterial expression vectors using ligase-independent cloning for protein expression and affinity purification to produce soluble substrates for in vitro reactions. To understand this topic, it's important to know what ribosomes are. Ribosomes are the universal machines responsible for protein production in all life forms. And if there are alterations to the function of ribosomes, it can have adverse medical effects like cancer and anemia. In humans, ribosomes are composed of 79 ribosomal proteins and 4 types of ribosomal RNA, which are synthesized individually and assembled into a complex. While we understand the mechanism of assembled ribosomal proteins, we know little about their interactions and modifications of individual ribosomal proteins. So during my research process, we separated the 2 sections into part 1 and part 2. Our part 1 was molecular cloning. Within molecular cloning, we had a few experimental approaches that we used, such as infusion cloning, negative selection, and molecular cloning. One of the first things that we did was designed a primer that overlapped part of the sequence in order to create a homologous region between the ribosomal protein gene and the expression plasmid. And then afterwards, we did PCR amplification, which allowed us to then separate the DNA bands by agarose gel electrophoresis. And then we did the linearization of the PNH TRXT vector, which was incubated with the PCR amplified target ribosomal gene. And afterwards, we did a hybrid recombinant DNA, which was transformed into stellar cells to seal the plasmid. Then we're able to basically generate the 8 recombinant plasmids. And that's what led us to part 2, which is protein expression. During protein expression, we had selected a few colonies for plasmid mini-preps and off-site sequencing in order to make sure that the fidelity of the recombinant sequence was correct. And we transferred the plasmids into BL21 cells, which allowed us to then add different concentrations to the IPDG to liquid cell cultures and do overnight incubations, where the cells were harvested by centrifugation, and resuspended them with lyse buffer. Afterwards, we separated the proteins into cell lysate by SDS Page and Kumasi blue staining, which allowed us to determine the presence of each ribosomal protein. In the future, we hope we can do other experiments such as affinity purification of the soluble ribosomal proteins using affinity chromatography, or using purified ribosomal proteins as substrates for in vitro reactions. Thank you so much. Hello, my